Hello students, good evening all of you. Welcome to Brilliant YouTube live classes. Today we are going to discuss one of the complicated chapter in biology, that is molecular basis of inheritance. Before that, let me introduce myself. My name is Sandeep. I am a senior biology faculty in Brilliant Doha, Qatar. So I just wanted to tell you something. From the next month, our annual exams are going to start. So our biology exam on 16th March, right? So students, we have very less time. So in this less time, we should cover up all the chapters, whatever the chapters we have, especially high weightage chapters. You know something? We have already discussed in our theory classes. The high weightage chapters are 3. What are the 3 high weightage chapters? Tell me. Principles of inheritance, molecular basis of inheritance, and evolution. And these 3 chapters, 20 marks. The highest weightage unit is genetics and evolution. In this unit, we have 3 chapters, right? These 3 chapters, especially from this 3 chapter, the second chapter, molecular basis, is very, very complicated. Many students are getting confusing in this chapter, right? So in this chapter, what the important questions are there, we are going to discuss. We are going to discuss from this chapter important and very, very complicated questions today. If you know the answers of these MCQs, kindly write your answer in chat box. Are you ready? Look at here. Look at the first question. What is the first question? Look at here. In this first question, read the following statements and select the correct option they are asking. We have to identify the correct option. Number 1, loosely packed and lightly strained region is chromatin or heterochromatin. Is it true statement? What is the meaning of heterochromatin? Tell me. Heterochromatin is that chromatin which is darkly strained and tightly packed. So what is here? It's giving loosely packed and lightly strained. So in place of here, I can write what? Tell me. It is euchromatin, right? This is euchromatin. So this is not a correct answer. This is not correct statement, right? Look at the next one. Densely packed, darkly strained region is called euchromatin is given. Exactly opposite is given, student, right? Exactly opposite is given. So first 2 statements are not correct, right? First 2 statements are not correct. Look at the third one. A typical nucleosome contains 200 base pair of DNA helix. Absolutely correct. Nucleosome. Shall I ask one small question? Nucleosome made up of? Nucleosome made up of DNA and also histone protein, right? DNA plus histone protein together called nucleosome. So here they given what? A typical nucleosome contain 200 base pair. This statement is correct. First 2 statements are wrong. So what is the right answer? Right. Look at the first one. Statement 1 and statement 2 are true. They given no. So look at the second one. Statement 1, statement 2 are false, right? 2 statements are false and the statement 3 is true. So what is your answer? The right answer is option, right? Tell me. Option B. Am I clear? First 2 statements are false and the last statement is 3 true. That is right. Very good. So tell me here, what is out of these 2, euchromatin and heterochromatin? Can any one of you tell me which of these 2 is highly transcription wise highly active? Highly active. Which of these 2 is highly active? Right. That is what? Euchromatin is highly active. Heterochromatin 1 is what? Inactive or less active, we can say. Right. Look at the next question. Next question. Next question. This question. Which one of the following makes use of RNA template to synthesis DNA molecule? RNA template to synthesis what? DNA molecule. Look at here. Which of the following makes use of RNA template to synthesis DNA? We know the central dogma. Can you please tell me what is central dogma? DNA, right? DNA to DNA replication. This is called DNA replication, right? replication. And DNA replication, what is the major enzyme? DNA dependent DNA polymerase enzyme. Fine. So DNA to the formation of mRNA, here we can write what? DNA dependent RNA polymerase enzyme, or simply we can write the RNA polymerase enzyme. And this process is called the transcription, right? This is transcription. mRNA to the directly formation of protein, this protein formation is called translation, right? This is transcription and this is translation. Fine. Look at the question they are asking. From the RNA DNA formation, from the RNA DNA formation, look at here, DNA polymerase enzyme, RNA polymerase enzyme, reverse transcriptase enzyme, and DNA dependent RNA polymerase enzyme. If you able to recall, student, central dogma in virus, RNA genetic material virus. What is RNA genetic material virus central dogma? Can you please tell me now? So RNA replicate, RNA replication. Tell me now, RNA to the formation of DNA. This process is called reverse transcription. Yes, students. Now tell me, DNA, from this DNA formation of mRNA, this is common process called what? Transcription process. And after that, protein formation. This is translation. Now tell me here, from RNA DNA is forming, so I can simply tell me reverse transcriptase. What is the other name of reverse transcriptase? Any one of you? What is the other name of reverse transcriptase? Allah, what is the other name of reverse transcriptase, Sania? It is RNA dependent DNA polymerase enzyme. Yes, student, yes or no? RNA dependent DNA polymerase. If from DNA RNA is forming, look at here, from DNA RNA is forming, our answer, the enzyme is DNA dependent RNA polymerase enzyme. From RNA, if DNA is forming, RNA dependent DNA polymerase enzyme. You understand my point? Now we can write what? Tell me, reverse transcriptase. Remember all of you, this reverse transcriptase enzyme specially present in the RNA genetic material. HIV virus also having this reverse transcriptase enzyme. That's why RNA will make a DNA, that DNA called cDNA, complementary DNA. One step extra in RNA containing viruses. What is the one step? Reverse transcription. Very good. Understand my point? Look at the next question. What exactly the next question? Next question, third question. This is a very easy question. I hope majority of students know this one. Can you guess and can you tell me the right answer? Come on, what is the right answer? Hafiz, Ala, Ganga, Sania, what is the right answer of this question here? The diagram shows an important concept in the central dogma, genetic implication of DNA. That is a central dogma. So we can write what? Here, DNA to DNA formation, it is a DNA replication. DNA to mRNA, just now I told you, what is that? The first one we can say transcription. So A is transcription, that is mRNA formation, and from mRNA protein formation, we can say simple B is translation. Who proposed this central dogma? Who proposed this central dogma? Look at here, Francis Crick. So the simple, that the question number 3 answer is what? Option A. Molecular basis of inheritance completely depending on this central dogma. How? From DNA to DNA formation, how? From DNA to mRNA formation, and what is the mechanism of protein synthesis. In this chapter, total chapter, we will discuss the synthesis of protein only. Understand my point? Look at the fourth question. This is the third question, very simple question. Look at the next question. Next question. Fourth question. This is a very easy question. There are special proteins that helps to open up DNA double helix in front of the replication fork. They are asking which enzyme will open the DNA replication fork. DNA replication starts from the particular area of DNA. Can you tell me the area? What is the area? For example, if you take, this is the double-stranded DNA, right? And this double-stranded DNA have a specialized areas. That specialized areas are called ORI. ORI stands for what? Origin of replication. Exactly to this origin of replication, one enzyme. What is the enzyme called? What is the enzyme? It is DNA ligase. DNA ligase enzyme join two DNA particles. DNA ligase enzyme join two DNA particles. It will not open up. So what is the right answer? It's not A. Come on, think all of you. DNA particle, this DNA ligase enzyme will join two DNA particle. So there is a one enzyme. Very good, Hannah. What is the enzyme? DNA gyrase. It is also known as DNA helicase enzyme. DNA helicase or DNA gyrase. This DNA helicase enzyme come and attach. This is called what? Tell me. Helicase enzyme. DNA helicase enzyme come and attach, break dash bonds. Any one of you, it breaks a particular bonds, and the opening of the two DNA strand. Which bond they break? Which bonds between these two DNA strands? Hydrogen bonds. They break the hydrogen bonds and the opening of this fork. This is called, tell me, replication fork. This we can write, tell me, this is replication. Yes, replication fork. Understand my point? Now, remember all of you, many times students will do a mistake here. You have to be read carefully. Are they asking about the replication process or are they asking about the transcription process? In case if they are asking about the transcription process, our answer would be completely different. You understand my point? So in this case, they are asking to opening of this ORI site, opening of this helica. That means what? The ORI site opening. Which enzyme? Helicase enzyme or gyrase enzyme. DNA ligase enzyme participate in two DNA molecule joining of two DNA molecule. And topoisomerase enzyme helps to remove the tension of DNA strand. Remove the tension of DNA strand. Next, DNA polymerase 1 enzyme. DNA polymerase, three different type of DNA polymerase enzyme, right? DNA polymerase 1, DNA polymerase 2. The important enzyme is DNA polymerase 3. The actual enzyme which polymerize the DNA. The actual enzyme which going to make the complementary DNA, that is DNA polymerase 3. DNA polymerase 1, removal of primers, filling the gaps. This is the function. So we can write the right answer is option C. Look at the fifth question. Fifth question I am going to show now. Now what is the right answer of fifth question? Can you do this? Any one of you answer this question? What are the structures called that give an appearance as beads on string in the chromosome when viewed under electron microscope? We have seen this. The length of DNA 2.2 long meter, 2.2 meter of long DNA. How come that long DNA embedded in a tiny nucleus? It is possible with the packaging of DNA, the DNA wrapping around the nucleosome, the DNA wrapping around the histone protein. As I know, student, when the DNA wrapping around the histone protein, it started to condense and they started to show the bead-like structure. The bead-like structures we can say nucleosome. Nucleosome. Look at here. Now, the nucleosome, I'm writing here. H2A, H2B, H2A, H2B, H3 and H4. And they're in the form of what? Tell me. Two are there. That's why we can say this is histone octamer. Can you please tell me, students? Pavitra, Ala, Ganga, can any one of you tell me histone protein made up of two different amino acids? They're rich in two different amino acids. Can you? histone proteins are rich in two different amino acids. Allah, Hiba, anyone of Samuel, which amino acids are present? Arginine and also arginine and one more lysine. Very good. So we can write this arginine and lysine, two are basic amino acids. So nucleosome histone proteins are basic in nature. Now here what happened? Which enzyme here? No, sorry, which one is going to wrap here? DNA. Look at here. DNA will come, wrap around and go. So this complete structure, which all present in this complete structure? DNA is there. Next, what also there? Histone proteins also there. So this histone protein plus DNA together we are calling what? Tell me. Nucleosome. Remember all of you, nucleosomes are rich in lysine and arginine amino acid. Nucleosomes are basic in nature. You understand my point? Now the next answer is, the right answer is what? Nucleosome. Look at the next question. Question number 6. This is a very simple question. Any one of you do this question? In prokaryotes, the genetic material, prokaryotes mean bacteria, Monera kingdom, bacteria organisms can come in bacteria, in prokaryotes. I have a question. Is in prokaryotes histone proteins are there? Pavitra, Sania, Hafiz, Samuel, histone proteins are there or not? I need answer. In prokaryote, histone proteins are absent. What type of DNA? Circular DNA. So we can write, tell me here, in prokaryote, like bacteria, what type of DNA? Circular DNA, right? Circular DNA. And without histone protein, histone proteins are not there. So I can take, this is, for example, if I am taking, this is a one bacteria, right? Now in this bacteria, there is a structure which is like this. This is called circular DNA, double-stranded circular DNA. I have question. In this is circular DNA. This circular DNA. Can any one of you tell me in prokaryotic circular DNA how many ORI sites are there? How many ORI sites? Any one of you? That means if it is a eukaryotic DNA, there are many Many ORI sites are there. From different ORI sites, the replication start. But in case of prokaryote, ORI sites are only one ORI site. Remember. So what type of DNA? Circular DNA, and no histone protein. Histone proteins are not there. So the right answer is what? Option B. Question number 6. Option B. Question number 7. You have to answer me. I am giving to the online student chance. You have to answer me. I will not explain. You have to answer me. What is the answer of this question? Question number 7. Read the very simple. Just now only I told this one. I will not explain once again. Can you tell me the right answer of this question? Nucleosome core made up of. Nucleosome core made up of. Answer. I need answer. Right, good, very good. What is the right answer? The right answer is Hiba, Pavitra, Samuel, and who is this? 1. Yes, tell me answer is, what is the answer of this question? Histone, nucleosome core DNA made up of H2A, yes, H2B, H3, H4, but they are 4 only, right? But we are calling the histone octamer. What is the meaning of octamer? 8 are there. So what is the, here only 4 are there. So 4, they are in the form of 2. So we can write, tell me, there are octamer. So there are 4 different type of, what are they? H2A, H2B, H3, H4. So what exactly the function of this H1? What is this H1? In NCERT, H1, one particular protein is given. H1 is the protein which holding these 8 octamer proteins. H1 protein is the one which is not considered as a octamer. No, which we will not include in the octamer. H1 protein is just for the holding of this 8 ball like structure of what? Histone octamer. In case of histomer, only 8 proteins are there. H2A 2, H2B 2, H3 also 2, H4 also 2. Remember, it's a simple question. Look at the 8th question. What exactly the 8th question? Can any one of you do this 8th question? 8th question. Read and give me the answer. Online student, fast. In DNA molecule, the one option is correct, the rest of 3 are wrong. Any one of you? Come on, answer me. Samuel, can you guess this answer? Allah. Now look at here. The proportion of adenine, A, that is purine, and relation to thymine, T, pyrimidine, varies with the organism. Varies according to Chargaff rule, same, right? Now there are 2 strands which run anti-parallel, one in 5' to 3' direction, right? Anti-parallel, one is always 5', okay? The opposite one will be now 3', right? Anti-parallel. So here I am writing what, tell me, 3', and this is what, tell me, 5', anti-parallel. Other one 3' to 5'. This statement is, how many of you tell this statement correct? Anti-parallel, one is 5 to 3, and one is 3 to 5. Yes, it's correct, right? Anti-parallel in nature, it's right. Now option C, the total amount of purine, Ag, total amount of purine, nucleotides and pyrimidine nucleotides not always equal. The way did Chargaff rule go? Where did Chargaff rule go? According to the Chargaff rule, purines are equal to pyrimidines, right? So next one. Last, there are 2 strands which run parallel. It's not parallel, it's anti-parallel according to Watson-Crick DNA model. So the appropriate right answer is good. Very good. The right answer we can say option B. Question number 9. 9th question, online student choice. Now look at the 9th question. 9th question. I hope you all understanding. Now, which of the following reunit? You have to tell me this one. Before that, to understand this question, we first you should understand splicing. Splicing. Let me write here something. Here, this is DNA. This is 3'. Okay. Now here I am writing the 3' to 5' of DNA strand, and 5' to 3' of DNA strand. Out of these 2, 3' to 5' we always call template strand, 5' to 3' we call coding strand. Yes, coding strand. This is DNA strand. Any one of you help me, any one of you tell me which out of these 2 DNA strand undergo transcription. Which of these 2 strand undergo transcription, coding or template? Template, right? Very good, it's template, right? Done. Now first, after transcription we will get hnRNA. This is hnRNA. This hnRNA has, tell me, coding, non-coding part. This is called exon, intron. Exon, intron. Coding, non-coding parts are there. Done? No, this one is called, I am writing E, exon. Exon is coding part, which is essential, participate in the polypeptide formation. Now this one is called what, tell me, intron. This is again exon, intron, exon, intron. So when exon, introns are present, this type of structure is called hnRNA. hnRNA. This hnRNA, we know it undergo a process called splicing. In splicing, what will remove? Which part of these 2 will delete? Any one of you? Intron or exon? Non-coding part. What is the non-coding part will delete? Non-coding part is called introns. Good, very good. Now introns will delete. So exon will be started to join. Wait 1 minute all of you. So when introns which are non-coding part, it delete. Now what is the next one is left? Tell me now. Here I am writing complete all small fragments of exon left. This all exon left. Now here I am writing this is also exon. This all are exon, all are exon only. I have a question, student. DNA, two DNA particles join DNA ligase enzyme, but here which fragments are joining? Which fragments are joining? RNA fragments are joining, right? You get my point? Are you with me? You get my point? Here RNA fragments are joining. So RNA fragments are joining by which enzyme? You have to find out now. Which RNA fragments are joining by? Look at here the options. RNA polymerase enzyme. RNA polymerase enzyme will synthesize the RNA, right? It will not participate in the rejoining. Look at the question. Which of the following reunit of exon segment? So I can tell the answer. Come on, any one of you. Any one of you read, right? Very good, absolutely correct. Who is this, Anya? Very good. So the right answer is what? RNA ligase. If in the exam question come two DNA particles are joining DNA ligase, if you are able to recall biotechnology chapter in the tools of biotechnology, we have seen this one. When vector DNA and target DNA, if you wanted to join DNA ligase enzyme, we are adding there. Done. DNA ligase enzyme we are adding there. But here when two exon, it is a single strand, RNA single strand. If you wanted to join this two DNA RNA molecule, you should use RNA ligase. So answer is what? RNA ligase. Question number 9, RNA ligase. Next question. Question number 10. Very complicated question. Any one of you? There are 3 codons which result in the termination of polypeptide chain synthesis. Translation, termination. There are 3 stop codons are there. Can you guess what are the stop codons here? Right? Look at here. Number 1, UAA stop codon, UAG stop codon, GUA. Can you tell me GUA code for? GUA code for, if I am not wrong, GUA code for valine. Am I correct? GUA code for valine. Am I correct? One of you, any one of you, Angeline, GUA, Alicia. GUA code for valine. Now look at the valine. Now here it is UAA. Yes, it is stop codon. UAG also stop codon. UGA also stop codon. So there are 3 stop codons are there, which terminate according to genetic code. Total 61 codons are there. Out of the total 61 codons, 61 codons are 64. Out of 64 codons, 61 codons are only code for all 20 amino acid. What about the rest of 3, sir? The rest of 3 are stop codon. They will terminate the DNA. They terminate what? Polypeptide synthesis. They stop the translation process. So 3 are stop codon. What are the 3 stop codon? You should remember this. Very easy. Look at here. UAA. First one. Look at here. UAG. Just bring this G here and UA here. UGA. Look at here. UGA. 3 stop codon which terminate polypeptide chain. Next. Look at the next question. Next question. Question number 11. Read carefully what this question is asking. A molecule that can be act as a genetic material must fulfill the traits given below except. This can come for 2-mark question also, not only for MCQ, 2-mark question also. Write the characteristic feature of a genetic material or any genetic material or DNA, RNA, if you are considering as a genetic material, it should fulfill 4 characteristic feature. What are they? Sir, it should undergo replication, it should undergo mutation, that mutation should be inherited, it should be constant. It should be constant, and number 4, it should be less reactive. Yes or no? It should undergo a Mendelian, what they call, characteristic feature. Now look at here carefully what they are given. It should be able to express itself in the form of Mendelian character. Absolutely correct. They are giving except. They are asking except. Number 2, it should be able to generate a replica. Yes, fine. It should be unstable structurally. No. It should be stable in structurally. If I am asking a small question, DNA, RNA, both are genetic material. The majority of organisms are DNA genetic material. Can you please tell me, out of these two genetic material, which one is more stable? Which one is more stable? Any one of you? Come on, student. Any one of you more stable? Pavitra, more stable. What is that? Yes, DNA is more stable than RNA. So RNA is, doesn't mean completely RNA is unstable. RNA is less stable. Now we can write what, tell me, except this character. What is that character, tell me? It should be stable. There are except. Right. Next question. Question number 12. This is lac operon concept I am giving to you. Can you solve this question, online student, whoever attending? Come on, answer me. It is a lac operon question, very important question. Lac operon, this question can come for 5 mark also from molecular base of inheritance. You should understand the complete lac operon expression, the regulation of gene expression in absence of lactose and presence of lactose. Look at here, a small segment is given here. Number 1, the given figure shows operon and its functioning. Select the option which is correctly able to A, B, X, Y, Z. A, B, X, Y, Z. Now first answer me. This diagram is in absence of lactose or presence of lactose? Absence of lactose or presence of lactose? Alisha, Sana, Hafiz. It is presence of lactose or absence? So it is absence, right? When absence of lactose, only one gene can give repressor protein. What is the gene? I gene. I'm a clear, I gene. Now tell me here, we can write. Now this I gene undergo transcription to give mRNA. This mRNA will give A. So what is A? So I can write A is repressor protein. What is this B? Inducer. So what is inducer here? Look at here, inducer here. Now this inducer is going to be attached here, right? Now if this absence of lactose, this repressor would have go and block what, tell me? Operator. Am I clear? So when operator block, RNA polymerase enzyme cannot slide over the XYZ. So there will be no beta-galactosidase enzyme, no permease, no transacetylase, right? Here inducer, I can tell what? Lactose. Inducer is what? Lactose. Now tell me here, A, I can write repressor, fine. B, inducer. Z, Y, A. Now here it is, we can write what? X, Y, Z. So X is beta-galactosidase, Y is permease enzyme, Z is transacetylase enzyme. So the right answer of this question is, we can write option A. Can you tell me which of this XYZ can break the glycosidic bond between glucose and galactose of lactose? Which of these 3 enzymes can break, or which of these 3 enzymes can digest that lactose into glucose and galactose? What is the enzyme? Yes, exactly. Very good. Beta-galactosidase enzyme. Remember all of you, beta-galactosidase enzyme is synthesizing. It's coming from the particular gene called LAGZ. This is called what? LAGZ gene. After that, Y permease. After that, transacetylase. Lac operon question, my dear student, before your final exam, lac operon thoroughly prepared. Who proposed this? What exactly lac operon? What lac operon is tells us? Okay, and gene regulation in eukaryotes. This is prokaryote. Lac operon in where? Expression of gene in where? Prokaryote, bacteria. But remember, this question can come for 5 mark, all of you. Remember, it's only MCQ I'm discussing, but this concept is important for 5 mark. Look at the next question. Next question number 13. What is the 13th question? 13th question. Can any one of you answer this question? tRNA, transferal RNA. Amino acid acceptor end of tRNA. Look at here. If you carefully read this one, tRNA, transferal RNA, right? Now here, let me take this one here. This is mRNA. mRNA, this is 5 prime, 3rd prime. For example, on this mRNA, AUG, UUU, UUC codons are there. 3 codons are there, which are representing 3 different type of amino acid. Look at here, what is coming? tRNA. Yes, this is tRNA. tRNA has 3 loops. Anticodon loop, which read this one, right? Which read codon. This is called codon, and this one is called anticodon. Anticodon. Fine. Look at here, whatever the small strand is there, this we can write what? 5 prime, and this is 3rd prime. 3rd prime always have a codon. We can say what? ACC. Exactly, here OH group is there. This place activated amino acid. My dear student, this is important. Activated amino acid go and attach to where? Go and attach to the 3rd prime of tRNA. Remember everyone, this type of MCQ can come. This type of MCQ can ask exactly which prime of tRNA the activated amino acid will come and attach. So this is 3rd prime. This activated amino acid come and attach here. Amino acid acceptor. How many of you correct this one? Very good, Yusuf. Who is this? Right, very good, Angeline. So the right answer we can say 3rd prime of tRNA. What my point? 5 prime, 3rd prime is 3rd prime. Exactly, they will what? Activated amino acid come and attach. 13th question. Next, 14th question. What exactly a 14th question tells us? 14th question. Very, very simple question. We discussed in lac operon just now. What exactly the answer of this question? Which enzyme will be produced when a cell there is a nonsense mutation? My dear students, nonsense mutation, frame shift mutation, missense mutation, there are different type of mutations are there. Let me take one small example to understand the nonsense mutation. Nonsense mutation means, remember, suddenly if a change, a mutation comes, it will lead to stop codon. It will lead to what? Stop codon. Let me take one small example. Look at here. I am taking one small example to understand this. For example, CGA, 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 CGA. This is DNA strand. This is template DNA strand. From this template DNA strand, what type of mRNA can come? Tell me here. G, C, it is U, right? GCU in mRNA. Now I'm writing this is mRNA. This is mRNA, right? GCU, 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 right? This is the mRNA can come. If look at here, any one of this place, especially here I'm writing, or one place, one nitrogen base, if it is changed to a what they call A, or any one nitrogen base is changed and leads into the stop codon. What is the stop codon here? If this complete one is going to be stop codon or UAA, or any one of the stop codon is forming with the new mutation, what happened? Normally synthesizing polypeptide chain suddenly will stop. Normally synthesizing polypeptide chain will suddenly started to stop codon. So it's called nonsense mutation. This mutation is called a nonsense. That means this DNA one place started to undergo mutation. That mutation leads to the formation of stop codon in mRNA. So unexpectedly stop codon comes. So here normally it is synthesizing polypeptide chain. Accidentally and what they call suddenly stop codon comes. It will terminate the polypeptide chain. But here the question is telling the nonsense mutation in Y gene. So what is the order of the genes here? We can write the order of gene is, what order of the gene of in lac operon? Tell me the order of gene in lac operon. I can tell. Now it is, first one is called lac Z gene, lac Y gene, lac A gene is there. The problem in this gene, here if it is a nonsense mutation takes place, tell me now here the Z gene is happily there, it's there. Z gene is not undergoing any mutation. Z gene is as usual it's working. Z gene is as usual it's going. So tell me which enzyme will synthesis if a problem in Y? So though no problem in the Z gene, sir. So beta galactosidase enzyme will come. So there is a problem in Y gene, the beta galactosidase enzyme normally. I told you wherever the problem comes, that gene only will affect. The previous gene will be proper only. So beta galactosidase enzyme simply it will synthesis. No problem if a nonsense mutation in where? Y. Remember nonsense mutation come because of DNA mutation in the mRNA. It leads to the formation of stop codon. So everything will be properly going, but suddenly stop codon comes, it will terminate. Remember this one, everyone. You got my point? It's called stop. What they call nonsense mutation. Next question. Next question. Question number 15. Question number 15. Name the enzyme that facilitate the opening of DNA transcription. Now answer me. DNA transcription they are asking. Just now only I told you if this type of question come. If this type of question come. What did I say? What did I say? Clearly I told you, you have to read the question carefully. If they are asking which enzyme opening the replication fork, helicase enzyme, gyrase enzyme, then which enzyme here? They are not talking about the replication, they are talking about the transcription. Any one of you? Which enzyme here? Which enzyme is opening the transcription? Which enzyme go and bind to promoter of gene? RNA polymerase. Samuel, correct? Yes or no, Angeline? Allah, answer me a face. What, which enzyme go and bind to promoter? RNA. So look at here, DNA ligase, DNA helicase, DNA polymerase, RNA polymerase. Tell me which enzyme is that? RNA polymerase enzyme go and bind to a promoter and start what? Opening of transcription. Got my point? In the transcription unit, we have promoter, terminator, template, coding stands, complete this transcription unit, right? Remember all of you. If a question come, they are asking to opening the replication for the DNA replication, our enzyme is different. If they are asking to synthesis the mRNA or transcription process, our enzyme is different. In this question, they are asking transcription one. So RNA polymerase. Can you please tell me how many RNA polymerase enzymes in eukaryote? In eukaryotic transcription, how many RNA polymerase enzymes are there? Is the same single, only one RNA polymerase enzyme? Or any different type of RNA polymerase enzymes? Answer me. Yes, good, Albert. How many? Very good. Three different type of RNA polymerase enzyme can transcribe 3 different type of RNAs. Remember all of you. So here it is, RNA. That's why remember student, read the question carefully. Are they asking about the replication or transcription? Look at the next question, 15th question, 16th question. 16th question, now you have to answer me. Now answer me. Look at here, everyone student. There are 3 different type of RNA polymerase enzyme and RNAs also. Write down here all of you. RNAs are 3 different type. First RNA is called what? Tell me. rRNA, ribosomal RNA, abundant RNA in animal cell. Number 2 is hnRNA, hnRNA, or I can write messenger RNA. Number 3, tRNA. Now, for the transcription of rRNA, RNA polymerase 1 enzyme. Remember student, RNA polymerase 1. For the transcription of hnRNA or mRNA, RNA polymerase 2, right? Now here I'm writing RNA polymerase 2 enzyme. Now for the transcription of tRNA, I can write RNA polymerase 3 enzyme. Based on this information, find out the right answer. Now I'm cross-check your chat box. Find out the right answer, students. Right. So first one is Hiba, RNA polymerase 1. RNA polymerase 1, sir, rRNA, ribosomal RNA, RNA polymerase 2, hnRNA, or I can also write here messenger RNA, right? RNA polymerase 3, tRNA. Sometimes you will get question like this. Sometimes you will get question what, for example, if you remove RNA polymerase 2 enzyme in the nucleus, which of the following RNA will affected? If you remove RNA polymerase 1, which of the following RNA will affected? Remember one more important thing, this all three different type of RNAs in the transcription, all three different type of RNA polymerase enzyme exclusively in where? Eukaryotic transcription only. In case of bacteria, in case of prokaryote, one single RNA polymerase enzyme can transcribe all the RNAs. So do not get confused. So the three different type of RNA polymerase enzyme for the transcription of three different type of RNA exclusively in eukaryote. So our final answer is what? 2, 3, 1. Where is 2, 3, 1? 2, 3, 1. So option C. Question number 16, option C. Question number 17, I'm giving to you. Question number 17. This is a repeating question, student. Everyone focus on the board. This question is often repeating in the competitive purpose and also CBSE. Carefully read the question what they're asking. Which of the following figures correctly represent the replication formed during DNA replication? DNA replication, when it's that means DNA will make DNA, new DNA always synthesis from which prime to which prime? New DNA always synthesis from. I need answer from you all of you. You should answer me. Can you send your answer in the chat box? Which prime to which prime new DNA synthesis takes place? Yes, very good. 5 to 3 prime. Look at here, all of you. New DNA always grow from 5 prime toward the 3rd prime. Even new mRNA also grow from where to where? 5 prime towards the 3rd prime. So look at here. First, option A. In option A, 3 prime to 5 prime the DNA is going. This is not correct. Arrow mark is going from 3rd prime to 5 prime it is going. I told you the new DNA will go from where to 5 prime towards the 3rd prime. Look at the option B. In the option B, okay, 5 to 3, this strand is correctly going. Okay, I can write this. Look at here, the second strand is 3 prime towards the 5 prime it is going, which is not correct. Okay, B also not correct. Keep aside. C, option C. In the options again from 3rd prime to 5 prime the newly synthesizing DNA is going. So it's not correct. Option D, if you look at in the option D, 5 prime to 3rd prime, this is 5 prime towards the 3rd prime is DNA going. Now from here 5 prime towards the 3 prime new DNA growing. So the right answer, remember all of you, sometimes you will get question like this, newly formed DNA or newly formed hnRNA, it grow from which prime to which prime? Our answer will be 5 prime to 3 prime. Remember, you got my point. So the right answer of this question is option D. Option D. Sir, it is DNA 5 prime to 3 prime. So why did you write the RNA also 5 prime to 3 prime? Because 3 prime to 5 prime. Because which one undergoing transcription? Tell me. 3rd prime to 5 prime of DNA. This is called a template. So template undergoing transcription, right? So if template undergoing transcription, obviously mRNA will be 5 prime to 3 prime, right? 5 prime to 3 prime, this is mRNA. So do not get confused. Newly synthesizing mRNA also 5 to 3 prime. Remember. Next question. Question number 18. What is the question number 18? This is a very simple one. Any one of you, this type of questions will come. Coding, standing, mRNA, template stand, coding stand, questions will come. Any one of you answer this question. I need answer from you all. Read the question. It is a particular sequence it is given. What is the sequence? Tell me. A, G, G, T, A, T, C, G, C, A, T. Can you please answer me which prime I should write here? Which prime I should write here? Which prime to which prime? Read the question carefully. In the question we given small hint. Hafiz, Ala, Angeline, Hiba, any one of you give me Albin, any one of you. First they given a small hint called coding strand already given. Yes, absolutely good. Who is this? Very good, Pavitra. Good. So they given what already in the question. If you carefully read what it is given, this is the DNA sequence from the coding is given. But coding means, so I can write 5 prime to 3rd prime. Answer me. 5 prime to 3rd prime of DNA is undergoing transcription. 5 prime to 3 prime is undergoing transcription. Answer me. 5 prime to 3 prime. No, sir. It is a coding. We need this. To get this one, we should transcribe the template, right? What did I say? This is coding. We need this. We need this codon. AU, AGG, TAT, CGC. This I need. So to get this codons, I should transcribe. What I should transcribe? Tell me now. So I am writing here. What is this? This is 3rd prime. 3rd prime. Tell me what is here? I should write here. It is T, C, C, A, T, A, G, C, G, T, A. This is which prime? Tell me. This is 5 prime. So here it is called coding strand. Fine. And this one is called template strand. Sir, answer got? Did we get the answer? No, not yet. Look at here. Template strand I am transcribing. Now this template strand I am transcribing. Come on, answer me all of you. Template strand. Now here I should write the 5 prime. Now tell me. A pairs with T pairs with A. C pairs with G. C pairs with G. A pairs with U. Why U? Sir, why did you write U? Because I am writing RNA. In RNA, T is not there. U is there instead of T. So, U. Okay. Now, T pairs with A pairs with again U. G pairs with C. C pairs with G. G pairs with C. T pairs with A and A pairs with U, and this is 3rd prime. So what? This is the mRNA. What is our right answer out of the 4 option? What is the right option? Very good, very good. Option. Tell me which is the right option. Yes, very good. Option A. Am I clear? Are you following me? Am I clear? Are you with me, students? Did you get my point? Option A is the right answer. Look at the next question. What is the next question here? Question number 19. Which of the following RNA should be most abundant? 1 minute. Which of the following RNAs most abundant RNA in the animal cell? Out of the 4 options, I already told you this one in previous question. Anyone of you answer me. Which is the most abundant one? Most abundant one is ribosomal RNA. Which out of the 4 is the smallest RNA? Which out of the 4 is the smallest RNA? Which out of the 4 is the smallest RNA? Answer me, Alisha. Good, very good. Smallest RNA is tRNA. Very good. Smallest RNA is tRNA, is also called adapter RNA. Smaller RNA, soluble RNA. This is also called soluble RNA. mRNA is called short short-lived RNA. Short-lived RNA. Remember all of you. Ribosomal RNA, abundant RNA, largest RNA. This we can also write what? Largest RNA. Remember all of you. Question number 20. 20, any one of you? Come on, ready to answer this question? DNA replication is very important question. Very important question. You have to think here. You have to think here before you give the answer. DNA replication. Come on, any one of you? Any one of you? DNA replication is conservative and discontinuous. Is this conservative or semi-conservative? 1 minute, student. Is it conservative or semi-conservative? I need answer. Is it conservative or semi-conservative? Michelson and Stahl's experiment, if you remember, sir, it is semi-conservative. Am I clear? Semi-conservative. So, semi-conservative and semi-discontinuous, semi-conservative and discontinuous. Out of B, C, one is correct answer. Anyone? Anybody? Anybody? Out of B, C, one is correct answer. Very good, very good. Who is this? Nitin, very good, absolutely. Here it is, semi-conservative and semi. Why we are calling semi-discontinuous? Because if you able to recall, there will be a one discontinuous strand is formed, right? It's called what? Come on, students. Discontinuous strand is also called something. I'm not seeing the message. I'm not seeing the chat box. Very good, absolutely Hafiz. Okazaki fragment. Okazaki fragment. That's the reason why semi-conservative, semi-discontinuous. Why we are called semi-conservative? Why we are calling semi-conservative? Because 50% DNA is the previous DNA, that means parent DNA. 50% DNA is modern DNA or new DNA. That's why we call semi-conservative. First time Michelson and Stahl conducted experiment in Escherichia coli with the help of N14, N15. Do you remember? And heavy nitrogen and light nitrogen, right? So we can write, very good, option B. 21st question. 21st question. I will cross check. Very simple. Everyone know this. Just now I told you. How many of you know this? Come on, answer me. DNA dependent RNA polymerase. DNA dependent RNA polymerase catalyze transcription one strand. Which strand is that? Which strand is that? We know this one. Which strand is that? Absolutely correct. Very good. How many of you giving answer? Right, very good. Angeline, right, but what is the answer of this one? One of the strand is template strand. So template strand start from 3rd prime toward 5 prime. So this will give mRNA. Remember. Next question. Question number 22. Question number 22. This question can also come from 3 mark. Write the complete structure of mRNA. Or here it is, transcription unit they given. Transcription unit, structure of complete mRNA. This all are very important concept. Now, given the diagram represent the compensate of the transcription unit. So what is A here? A, we can write simple P. What does P stand for? Promoter. Right, promoter. And B stand for what this is? Terminator. And C, D, you have to tell me. You have to tell me. C, D, yes, absolutely, very good, absolutely. So we can write what, tell me. So one is called promoter, terminator, coding and template. Where is the coding? This is a C, actually it says C is template, right? Template and also coding. So we can write the answer is what? Yes, option. Is it correct? Option C is correct, right? Very good, absolutely. So these are few important questions from molecular base of inheritance, and if you need a theory of any one of this topic, you can go to Brilliant YouTube channel. It will be clear, description is given, and with the new chapter next class we will discuss few more questions, and try to follow this channel, and this session will, I hope, it's help you to get a good marks in molecular base of inheritance. Thanks for attending Brilliant online class. Thank you.